The neo-Darwinists attempt to explain everything with the concept of adaptation, but today we know this is not the case. The neo-Darwinian literature completely lacks critical sense. They provide us with implausible answers, and the entire theory falls apart. There's an uproar if Darwinism is challenged. Organisms do evolve, but Darwin's theory is not at all coherent.
These are the words of American philosopher Jerry Fodor, who critiques Darwin's evolutionary theory and its continuation by neo-Darwinists with precise arguments. As many of you are aware, we have discussed the theory of evolution multiple times, highlighting its unclear aspects and doubts. Today, we'll approach the topic from a genetic perspective, delving into some obscure points of our genome and fearlessly exploring new and surprising scenarios. DNA, the instruction manual that distinguishes humans from every other species on Earth, hides something mysterious. Something that might reveal an origin different from what we've been told over the years. Before embarking on this fascinating journey, as we always do, we invite you to subscribe to our channel to support our work and to stay updated with our new videos. Mutations are the basis of all evolutionary processes. Indeed, the DNA in our cells is constantly mutating, a normal physiological process due to the error rate that occurs during the replication phases by enzymes in DNA duplication. It is estimated that every day thousands of our cells undergo spontaneous mutations. Of course, to these mutations, we must add those that occur when exposed to mutagens, such as UV rays. However, the only mutations that can be passed on to offspring are those that occur in germ cells, in oocytes in women, and in sperm in men. Researchers have estimated a rate of about 60 mutations per cell generation in this case. This is still a very slow process that, on one hand, underlies all the genetic diseases we know today, and on the other, is the engine of evolution as we know it today. These continuous mutations, passed down from generation to generation, contribute to an inexorable deterioration of our genome. Before proceeding further, let's make a promise. From now on, we will tackle very complex topics, but we will do so in the simplest way possible so that everyone can understand the mysteries hidden in our DNA. Following this promise, let's imagine our genome as an encyclopedia composed of 23 different volumes, the volumes being our chromosomes. Of each volume, we actually possess 2 copies, which are similar in that they contain the instructions for building a human being, but at the same time different, as each pair of chromosomes is composed of 1 chromosome inherited from the father and 1 from the mother. Every cell in our body potentially has access to this entire set of information, but in reality, they can only read a small part of it, depending on their specialization. For example, a heart cell and a neuron only read certain books from our encyclopedia, specifically those related to their functioning. This naturally raises the question, how do cells read the information present in DNA? To be deciphered, the information is transcribed into RNA molecules, which are then translated into proteins, the actual functional molecules in cells. Here arises the first fascinating mystery. Our DNA is composed of segments called genes, which can be either coding, meaning they contain instructions for protein synthesis, or non-coding, which do not have this property. Until some time ago, scientists believed that the only genes worth considering were the coding ones, those capable of transcribing DNA into mRNA molecules and then into proteins. However, there was a curious fact they were still unaware of. The coding DNA segments that make up the tomes of our encyclopedia represent only 2% of all DNA, leading to the question of what purpose the remaining 98% of our genome served. Initially, it was thought that this did not have any specific importance, that it was just the result, or rather the proof, of all the discard processes due to evolutionary processes, and it was thus called junk DNA, to indicate its perceived irrelevance. However, this classification was short-lived. To the researchers' surprise, simple beings like the Caenorhabditis elegans, a small worm living in the mud, actually had more coding DNA than a human. How could such a simple and elementary being have more coding genes than a complex being like a human? Clearly, something was being missed in this analysis. It was only later understood that the role of junk DNA was far more important than initially thought, although some aspects remain mysterious and may be key to explaining the mystery of humans. But let's set this concept aside for a moment. We'll return to it later in the video. Now that we've understood how DNA is structured, we can analyze a first anomaly present in our evolutionary path. Currently, we have sequenced the genome of two of our distant cousins, specifically Neanderthals and Denisovans. Unfortunately, the quality of the genetic material is not such as to allow a precise comparison with human DNA. The only way to attempt a comparative study is to analyze the DNA of our still living distant relatives, the chimpanzees. Comparing the two genomes, it was found that they differ by 5%, so this small percentage is what significantly differentiates us from apes. But the comparison between the two DNAs brought to light a detail that is still the subject of study and debate. A significant difference between us and the great apes is found in the number of chromosomes. As we mentioned at the beginning of the video, human DNA is composed of a pair of 23 chromosomes, 46 in total, while that of chimpanzees consists of 24 pairs, 48 in total. Specifically, scientists have highlighted that in monkeys, the chromosome pairs 2A and 2B in humans have fused to form chromosome 2. This fusion is known as Robertsonian translocation. In humans, we witness this kind of event when chromosome 14 and 21 unite, as occurs in individuals with Down syndrome, also known as trisomy 21. Because the individuals in question possess two normal copies of chromosome 21 and an extra supernumerary copy of the same chromosome 21 attached to 14. Thus, they have one extra chromosome. Well, if this event is rare yet still possible, what exactly makes the fusion of chromosomes, an event that occurred millions of years ago and definitively distinguished hominids from other primates, so mysterious? The answer is simple. Individuals with aneuploidy, that is, those possessing one or more extra chromosomes, suffer from infertility. To be more explicit, molecular biology does not allow the creation of a new individual from the union of two gametes, sperm and oocyte, that have a different number of chromosomes, as it considers this event an error that cannot perpetuate. So to recap and clarify the situation, millions of years ago, at an unidentified moment, a primate in which the fusion between chromosome 2A and 2B into a single chromosome had occurred, incredibly encountered another individual of the same species carrying the same aneuploidy, and their mating gave rise to a new species. Why do we speak of a new species? Because, as we said earlier, nature does not permit the union of two germ cells that have a different number of chromosomes, recognizing this eventuality as an error. Thus, the newborn from two parents with one less chromosome will no longer be able to mate and produce offspring with all individuals who have an extra chromosome. This is why a hominid of the Homo sapiens species cannot produce offspring by mating with another primate, a monkey to be clear, although attempts have been made in the past, particularly by Russian scientist Ilya Ivanov, who inseminated a female chimpanzee with human semen but achieved no result. What happened millions of years ago in a remote part of Africa is therefore undoubtedly a very rare event, and the very low probabilities of this event happening and giving rise to our species have raised several doubts. To this must be added a further detail. The analysis of the chromosome in question, once examined, would indeed have raised further doubts.
Apes have 48 chromosomes, but humans have 46, so evolutionists have a discrepancy in chromosome numbers and need a solution that somehow makes us more closely related genetically. They believe two ape chromosomes fused end to end and formed a single chromosome, human chromosome 2. They actually found a sequence that appeared to support that idea. There's a stretch of DNA with some telomere-like sequences, like the ones found at the ends of chromosomes in the middle of chromosome 2. They called this the fusion site. In a chromosome fusion, there should be two genetic scars, and you should have a fossil centromere in addition to the fusion site. As it turns out, the so-called fossil centromere is also in the middle of a gene, a huge protein coding gene. This alleged fossil would be impossible to form in a fusion. So fusion is debunked.
In this rare and improbable event, some researchers see proof that humans and monkeys do not share a common ancestor, while others see it as a sophisticated act of genetic engineering, performed by an external entity that somehow selected the human species, forever distancing it from its simian progenitor. Certainly, if it were a natural event, we are talking about something extremely rare and, in some ways, bizarre. Bizarre because all known conditions of aneuploidy in humans are rarely compatible with life, and when they are, lead to pathological conditions, such as in the case of trisomy 21 or Down syndrome. We must add another detail. We do not know in practical terms what the fusion of these two chromosomes entailed, what traits this event endowed the new species with, but it is highly unlikely, if not impossible, that this alone determined the unique characteristics of humans. The only thing we are certain of is that this has forever separated humans from all other simian beings, distancing them into a new species. Now, whether all this is the fruit of the improbability of chance or an external intervention remains a fundamental question to answer. What made humans the unique beings we know? Answering this question is not easy, but analyzing our DNA presents a very interesting hypothesis. The answer to this question might in fact come from an event outside our planet. But let's proceed in order and revisit the concept we talked about in the first part of the video, referring to coding and non-coding DNA. As we said, most of our DNA is non-coding and was long considered useless. But in what was defined as junk DNA, specific genes called jumping genes or transposable elements were identified, exhibiting very peculiar characteristics. These DNA sequences have the ability to duplicate and copy themselves in different parts of the genome through a mechanism of retrotranscription. In simple terms, it's like having within the DNA sequences that occasionally decide to create a copy of themselves in the most desperate parts of the genome, modifying it. These retrotransposable elements are considered responsible for the most significant rearrangements our genome has undergone, as well as for the expansion of non-coding DNA. To simplify, we can say that these elements have the incredible capacity to shape our DNA, bringing out new genes from unused DNA strands. However, these incredible elements, after participating in the creation of the human project, deactivated themselves, accumulating mutations that switched them off. But there are methods that can reactivate these little engineers and awaken them from their genetic slumber. Currently, the only place where they can awaken is in neurons. And when this happens in an individual, neurodegenerative or neuropsychiatric disorders develop. However, studies on rodents have shown that the reactivation of these retrotransposable elements can also bring benefits or specific peculiarities, such as the formation of long-term memory. The stimuli capable of reactivating them seem to be environmental stress, high temperatures, and ionizing radiation. To clarify the concept, let's use the encyclopedia example and pretend that our coding DNA is all the text present in the tomes that make up our volumes, and that our non-coding DNA are billions of seemingly meaningless words. These particular genes have the ability to take individual words and insert them into our volumes. Once inserted, these modify the sentences, giving them new meanings. If we add that the behavior of these genes can be influenced by external events, undoubtedly fascinating speculations open up. Is it possible that our evolution has been somehow guided by events we are currently unaware of? 2 million years ago, around the time of a presumed diversification among hominids, a star exploded relatively close to our planet, inundating it with radiation. Could these radiations have influenced our evolution? And if somehow this event gave a push to human history, triggering those changes in our DNA that realized the peculiarities distinguishing our species, why didn't it affect all other living beings on Earth? Species jumps. The mystery of the Cambrian explosion, the sudden flourishing of countless life forms, the evolutionary anomalies found in research. Could all of this have been triggered by an external event that provided the impetus for all this to occur? These are all fascinating questions, especially now that we have discovered another aspect that has revolutionized our understanding of DNA. As we were saying, the way coding DNA transcribes information works like this. Genes encode DNA into RNA, which is then translated into proteins. Well, it was thought that this was the only way to create information, and that it was a mandatory step, so much so that it took the name of the central dogma of molecular biology. But in reality, we have discovered that things are not so straightforward, and that the reverse process can also occur, whereby strands of RNA can become DNA. This axiom is very important because it is one of the foundations upon which the entire evolutionary theory is based. A random event that occurs during the transcription of genetic material gives rise to a mutation, which then determines a trait that may favor evolution. But if we have reverse transcription events, that is, RNA molecules turning into DNA, it means that not all genetic changes occur through random mutations, but these can be generated by the genome itself. The genome is thus capable of modifying itself and giving rise to new traits. If we accept this, however, all the premises upon which evolutionary theory is based would fall, and we would then have to ask, what is DNA? The answer could be that it is a code, the creative matrix of all forms of life, a huge encyclopedia in which all possible forms of life are written. A code subject to random mutations, but which can at the same time intervene on itself. Wouldn't the impossible improbability of all those events that dot the evolution of life become less improbable if we admitted just for a moment that the building blocks of life followed a predetermined path? And if we accept this, what is this external guidance? Is it information traveling through the universe in the form of energy? Information that originated with the very origin of the universe? At the moment, we have only scratched the surface of a mystery that envelops not only the origin of man, but life itself. Within us may lie the answer to all our questions. We might finally explain the enigma not only of our origin, but of life itself. An enigma that seems more and more each day to bind us to something alien that permeates the entire universe. But this is another story that we will tackle soon. For now, let's stop here. We are really curious to hear your opinion. Let us know in the comments what you think. And if you liked the video, don't forget to subscribe to the channel because your support is essential for us. See you in the next story.